First speaker is Zahid Siraj, who will be talking about SABRE-Enhanced Benchtop 15N NMR and efforts toward synthetic N15 labeling of pyrazinamide, an antibiotic and potential MRI contrast agent. Please, Siraj, the screen is yours.
Okay, thank you so much. Thank you so much giving me the opportunity to deliver my talk. My name is Zahid Siraj and I'm working in Dr. Goodson lab and at SIU Carbondale. So today I'm present my talk on pyrazinamide. Pyrazinamide is an FDA approved antibiotic drug, primarily used for the tuberculosis, and it can enhance the radiation response in hypoxic tumor. Besides this, it also possesses the cardioprotective properties. It can be used in the treatment of heart diseases. Cyber hyperpolarized could enhance PGA NMR detectability. So based on this approach, it could be used as a hyperpolarized PGA variant as MRI contrast agent. So our goal was to create a SABRE hyperpolarized pyrazinamide, which is safe, low-cost, biocompatible MRI contrast agent for biomedical application. So this is the signal amplification by reversible exchange. We adopt this methodology and we use the iridium-based catalyst, and iridium accommodate the parahydrogen and the pyrazinamide, and the spin was transferred successfully from parahydrogen to the pyrazinamide, and eventually we got a hyperpolarized substrate. And the source was the parahydrogen and the catalyst was iridium, and the target was an hydrocyclic compound, which is pyrazinamide in that case. So first we did the hyperpolarization study on the proton. So a proton on the pyrazinamide. So we give the color codings of all these proton to make the understanding easy. So you can see the proton, which is red and blue in color, enhanced more than 100-fold, while the one which is in the green in color, is enhanced a little less than the 100. So the reason is because the green proton is far away from the binding side, and it's also the sterically hindered side. So that's why it is assumed the enhancement is low in that case. So after the signal enhancement on the proton, we try to do the characterizations of the pyrazinamide. We try to find out the best pyrazinamide field. And the good news is we got the 5.5 millitesla, which is close in agreement with the previous paper in which they did the proton study only. And right after that, we try to find out the build-up time, which is in between 16 to 18 for all these three proton. And then we try to find out the signal decay, and the decay time was in between 16 to 20 seconds at high field, which is 9.4 Tesla. And when we did the signal decay at the low field, which is 0.55 millitesla, the decay time in between the 17 to 23 seconds. So if you see the enhancement on the green proton is low, the reason is again the same, because the green proton is far away from the binding side, which is that one, and it is also the sterically hindered. Next, we did the proton. We tried to image the proton on the low field proton MRI. And these are the two figures. This is the low field proton MRI on which we got the enhancement, and the make is 1.6. But because our contact with the vendor was ended, and so we trade in that MRI with the new one, and the new MRI we have is the make is 1.9. But we got all the data on the old one, which is 1.6. The strength of the magnetic field is 64 millitesla. So this is the schematics of the SABRE. setups, and I'm thankful to one of my colleagues, Nadia, who make this picture, and the paper is already out and is published in the Journal of Analytical Chemistry. So if you can see here, these Falcon tubes contain the water sample, the mixtures of the H2 and the D2O samples. And in the middle, we have a substrate, which is pyrazinamide. So now the question is, can we successfully image the pyrazinamide with low field clinical MRI scanner? So let's try to find out the answer for that. So if you can see the varying intensity of the white color. So these are all the water samples with the different intensity of the mixtures of H2 and the D2O. And in the middle, we have a pyrazinamide sample. But when we put all the setup into the MRI scanner, and we didn't use the parahydrogen bubbling, so we didn't get any signal. So there is no signal from the pyrazinamide. But when we did the parahydrogen bubbling for 30 seconds, so with the flow rate of 20 SCCM, so we got a signal from the pyrazinamide. So it's mean we successfully image the pyrazinamide in low field proton MRI. And after that, because the images were stored as a DICOM images on the hyperfin clouds, after that we downloaded these images on our local PC, and then we processed these images on OpenCV in Python, and we got the enhancement, which is more than 1,000-fold. So we successfully proton images. We did the successfully images with the pyrazinamide and with the low field proton MRI. After that, we did the N15 hyperpolarizations by using the SABRE sheath. So why we use the N15? Because it's a rare isotopes, no background signal, and the long polarization time with very sensitive chemical shifts. So first we did the N15 enhancement in our labs, and the detection field we use is 9.4 Tesla. And we successfully got the signal, and the enhancement where we calculated it was more than 4,000-fold. So this is a good news, we got a signal. But the bad news is, because our systems was not optimized, and the signal to noise ratio was too weak, so it didn't allow us to characterize or measure the T1 of the signal. Then after that, we tried to find out the solutions, and we did the hyperpolarizations of N15 on more optimized systems, which is 1.4 Tesla. And when we did the hyperpolarizations on that field, so we got a very strong signals. And when we calculated the signal enhancement, it was more than 140,000-fold at 1.4 Tesla. And but we got only the one big peak that demonstrate only the one nitrogen is attached with the iridium catalyst, while the other catalyst is, we got the peak. This may be associated with that nitrogen or is weakly binded, but we are not sure about that. But of course, we got one big signal from that nitrogen, and the enhancement was pretty good. And then after that, we tried to do the N15 SABRE sheath optimizations and the build-up dynamics for the PZA. And the activations time for the catalyst was 30 minutes, although most of the catalyst was activated in 10 minutes. Right after that, we tried to find out the best magnetic field. And the best magnetic field we got is 0.5 microtesla. And right after that, we tried to find out the best temperature times. And the best temperature for the hyperpolarizations of PZA was a little above than the room temperature is 27 degrees centigrade. And the build-up times is 9 seconds for the N15. So after that, we did to try to do some other parameter as well. We try to find out the best flow rate times, and flow rate, the best flow rate was 90 SCCM. It was calculated, and the concentration was a little above than the 100 millimolar, which gave us the best enhancement, while the signal decay, or the T1, is at the high field, which is 1.4 Tesla. In that case, is the signal decay was 129 seconds, while at the low field it was 21 seconds. So it's mean we successfully hyperpolarized the N15 on the natural abundant, and we got the signal, and the decay time was more than 2 minutes. And the paper is all these data we have already published in ACS Physical Chemistry Journal B. And right now we are also working on the synthesis on the N15 label of pyrazinamide, because it is a very small structure molecule. So first we try to adopt the zinky salt strategies or methods from one of the old paper, and we try to synthesize by using this scheme. But after a couple of attempts, we were not successful in getting the labeled pyrazinamide. So the reason is because the zinc salt is effective in case when there is a one nitrogen in the heterocyclic ring. But in our case, in the pyrazinamides, we have the two nitrogen in the rings. So that's why it wasn't successful. So we try to then adopt another alternative approach, and we try to synthesize it from the beginning. And we use the N15 glycine for that. And right now, after because it's a very long scheme, but right now we are on the second last step, is we do hope in the future we will have labeled pyrazinamide. We successfully insert the N15 into the heterocyclic ring of pyrazinamide. So in the summary and the future direction of HP pyrazinamide is we successfully a, a hyperpolarized MRI contrast agent, which is pyrazinamide using proton saber and the saber sheath for N15. Proton spin of PZA was easily hyperpolarized with saber at 5.5 millitesla, while the proton enhancement we got is more than 100-fold at 9.4 Tesla. And hyperpolarized PZA was successfully imaged with the low field clinical MRI scanner, and it gives the enhancement is more than 1000-fold. And also the direct N15 hyperpolarizations of naturally abundant N15 spin is possible by using the concentration 50 millimolar, and the detection field was 9.4 Tesla, and the enhancement we got is more than 4000-fold. By improved setup, this enhancement and the signal was very high, and it was about more than 140,000, while the detection field was 1.4 Tesla. And the signal decay over the lifetime of the signal was more than 2 minutes. Currently, we are working on the synthesis of the N15 label PZA that ultimately allowing studies in cell cultures. And I'm very thankful to my PhD advisor, Dr. Boyd M. Goodson, and the Goodson lab members, and also my collaborators, our collaborators, but specifically Dr. Edward Chekminov at Wayne State in Michigan. So he allowed us to do the N15 hyperpolarizations at 1.4 Tesla. And also, I'm very thankful to the funding agencies, NSF, NIH, DOD, and SIU, GAVA and DRA fellowships. So this is all from my sides, and thank you so much for giving me the opportunity.
Thank you for staying on time. We have time for maybe one or two short questions. So the first question from Andrei Pravdivtsev in the chat. Why the T1 is so fast at low fields and build up even faster compared to high field T1?
For proton, you're
I think, I don't know. I think it was about N15. Yes, Andrei just.
here. Okay, so because at high field, this is we placed our hyperpolarized PZA in 1.4 Tesla, and that we just measure the times in high fields. But in the case of the 0.4 microtesla, we first bubbling in the 0.4 microtesla, and after that, because we shift from 0.4 to the 1.4 Tesla, this is maybe the one of the reason. The second reason is because at 0.5 microtesla, the frequency difference of the hydride is matches with the J coupling more efficiently, and the polarization transfer was happened efficiently at that low field. But at the high field, like 1.4 Tesla, maybe the other factor like the frequency difference of the coupling span is higher than the J coupling. This is maybe the reason, and maybe the is not very high field 1.4 Tesla, but maybe the chemical shift anisotropy play any role in that one, which is the alignment of the spin relative to the field.
Thank you. Thank you for your answer. And I think in the interest of time, we will move to the next speaker. But of course, questions can be continued to be typed in the Q&A box in
Okay.
Zoom.
Thank you so much. Thank you so much.